Okay, so this is chapter 15, part 5, and in this part of the chapter, we're going to focus on ribosomes and translation and protein synthesis. And this is the end of the chapter. Okay, so let's talk about ribosomes first. So ribosomes are going to be the site of translation. So they are what is doing the translation. So again, they're going to read mRNA, and as they read the mRNA, they're going to put amino acids together to form a polypeptide, a protein. What are they made of? So they're made of two subunits, and those two subunits are both made of protein and ribosomal RNA. So that's that rRNA. And the two subunits are named the small subunit and the large subunit. And they're named that way because of their size. So that's nice. What the ribosome is going to do is it's going to attach to the 5' end of the mRNA, and it's going to travel along the mRNA, reading the mRNA codon by codon, so 3 bases at a time. And it's going to allow the tRNA molecules with their attached amino acids to come in and start to put together those amino acids in the proper order according to the codons found in our mRNA. And so the ribosome, we say, is translating the message into proteins. Okay. Okay. So let me show you what a ribosome looks like. Okay, so I've got a couple of different pictures here. Again, I kind of like them all. They all show this a little bit differently, and they all show it well differently. So I'm going to start with this. So this is what an intact full ribosome looks like. And so you can see it's a kind of a globular structure. It's made up of a large subunit, which is shown here on top, and a small subunit, which is shown here at the bottom. And there's this little groove between them. And that groove between them is where the mRNA is going to be pulled through. Okay? So if we were to just take those apart, the small subunit looks like this, and the large subunit looks like this. And there's actually 3 sites in the large subunit. They're called the E site, the P site, and the A site. Okay? And I actually want to come over here and look at this ribosome, put together. It actually shows the groove right in between the large and small subunit is where the mRNA is going to be held and be read. Inside of our large ribosomal subunit, again, there's the A site, the P site, and the E site. And let me make sure I say this correctly. So I usually when I read this, I read it from right to left, the opposite of how we would usually read it, because this is kind of where it starts. And so let me talk about the A site first. So that A of the A site stands for the amino acyl site. This is where amino acyl tRNA molecules, so tRNA attached to an amino acid, this is where they're going to enter the ribosome. Okay? And they're going to enter the ribosome in this site, and their anticodon is going to complementary base pair to the codon in the mRNA. Okay? Once that happens, the ribosome is actually going to shift. And so now in the P site will hold the amino acyl tRNA that was just in the A site. Okay? And the ribosome shifts to the next codon, so the A site will be open again, and the next amino acid comes in. The amino acyl tRNA, I should say, comes into the A site. So the A site is like the acceptor for the amino acyl tRNA. The P site is going to hold tRNA molecules and the growing polypeptide chain. I'll show you this in just a second. So this P stands for peptidyl site. And then E, the E of E site stands for exit. And so once the tRNA molecule makes it to the E site, it's done doing its job and it can just leave the ribosome. Okay, so let me show you this in a different way. Okay, so same idea, just a different figure. And I like this figure a lot because it shows tRNA in every site, which is nice. So the ribosome looks a little different, right? It's shown here in gray. It's still made up of the small subunit and the large subunit, and there's still the A site, the amino acid site, the P site, the peptidyl site, and the E site, the exit. And you can see where they are attached. There's this groove, which is where the mRNA is going to be pulled through. Okay? Here is the process in full effect. Okay, we're not just looking at the ribosome, now we're looking at all of the parts and pieces. So in orange here, we have our mRNA that's going to be read and translated. So this is the 5' end, and if we move down it, this is the 3' end. Okay, the A site again is going to accept amino acid tRNAs. The P site is going to hold tRNAs with a growing polypeptide chain, and the E site is where those tRNA molecules are going to exit. So let me start back over at the A site, and let's take a look here. So in the A site right now, the amino acid tRNA just came in. And let's look at the mRNA. The mRNA reads GGC. The anticodon on our tRNA is going to be complementary, so CCC. So we can see the complementary base pairing that's happening right here. Now the codon GGG, if you flip back in your previous chapter notes, you'll see if you find your genetic code table, GGG codes for glycine. And so the ribosome is going to bring in the right tRNA, right, that has the complementary anticodon, carrying the right amino acid according to the message into the A site. Okay, what's going to happen next is this entire growing chain of amino acids is going to be transferred over to glycine. Okay, so this phenylalanine is going to form a peptide bond with this glycine that's catalyzed by the ribosome. What will happen next is the ribosome shifts, and this tRNA molecule will end up in this site, carrying this growing chain of polypeptides. This tRNA molecule that now will not have these amino acids attached to it moves into the E site, and it can then leave. Okay, so during this process, the tRNA molecule in the A site ends up getting all the growing chain of amino acids added to it, and then it shifts over to the P site. The next one comes in. The cycle continues. Okay, so we have kind of a general idea of how this works. So let's talk about the steps. So whether we're talking about prokaryotes or eukaryotes, it's the same 3 phases with, again, some subtle differences that we'll mention. The 3 stages are nice and easy, just like we saw in transcription. For translation, it's also going to be initiation, elongation, and termination. So the steps stay the same. Okay, so remember, we're going from mRNA to protein here. In translation, transcription, we went from DNA to RNA, and now in translation, we're going to go from RNA to protein. Okay, so this is initiation. So let's get our bearings here. So we can see 5' to 3'. This is our mRNA transcript. Okay, this is the small ribosomal subunit here attached to our mRNA. It also has these 3 little spheres, which are called initiation factors. And it's lined up so that here is the start codon, right here, reading AUG. That's the initial, the universal start codon. Upstream from the start codon will be something called the leader sequence. And this leader sequence is, in prokaryotes, is going to help the small ribosomal subunit find the start codon, find the gene, okay? So that happens first. So the small ribosomal subunit with its initiation factors binds to our mRNA, travels down it until it finds AUG, which is the start codon. Once it finds AUG, it's going to bring in the initiator tRNA, which is always going to be a tRNA attached to methionine. In prokaryotes, the methionine is going to be formulated, so it's called FMET. This is our first amino acid. So we have our tRNA molecule that is complementary base pairing to our start codon. That's going to trigger the release of one initiation factor. Then the large ribosomal subunit comes in and binds, and that triggers the release of the other initiation factors, and that is our initiation complex. Okay, so we have our mRNA, we have our small ribosomal subunit that binds to the leader sequence, makes its way down to the start codon. That's going to bring in our first amino acid tRNA, which is always methionine. In prokaryotes, it's formal methionine, or FMET. That's going to trigger the release of initiation factor, which brings in the large ribosomal subunit, and the rest of the initiation factors come off, and that's it. That's initiation. Okay. Again, in prokaryotes, the methionine of our initiator tRNA is modified. We call it FMET. In eukaryotes, it isn't. So it's just methionine, or just MET. Secondly, in prokaryotes, this leader sequence, it's actually called the Shine-Delgarno sequence. It's a conserved sequence upstream from the start codon. In eukaryotes, it's just a different consensus sequence, and it's actually different depending on the gene. But again, it's going to be near the start codon, which will help our ribosome find that start codon, the AUG. Helps it to find the gene. Okay. Lastly, the initiation complex in eukaryotes is much more complex. So instead of just 3 initiation factors, there's, upwards of 10. Okay, but that is initiation in a nutshell. Okay, the second stage of translation is elongation. And elongation in prokaryotes and eukaryotes looks exactly the same. And so it's just going to be this repeated round after round of the appropriate amino acid tRNA coming into the A site, its anticodon complementary base pairing with the codon of mRNA, and then the movement of the growing polypeptide to the amino acid in that A site. Okay, there are helper molecules that help this process along. It does require what are called elongation factors, and it also requires energy, right? Because we're building a larger molecule here. So again, we're simplifying it, but in the back of your mind, remember that it's a little bit more complex. And so here we go. Here's a nice elongation picture. It's the same one that we actually just saw. Okay. And so again, the ribosome just chugs along the mRNA, reading it 3 bases at a time. And for every codon, it's going to bring in the appropriate amino acid tRNA. It will complementary base pair. It will then shift, right? This growing polypeptide chain gets transferred. And then this shifts. This amino acid tRNA would be in the E site. It will leave. And the cycle continues until termination. Okay, so how does termination work? So the ribosome is going to keep moving, right, and keep adding amino acids, keep bringing in amino acid tRNAs into the A site and growing that polypeptide chain until it reaches a stop codon. Remember, there's 3 stop codons. Go back and look at that genetic code if you need to. If any of those 3 stop codons are in the A site, that's it. We're at the end of the coding sequence. And so a protein called the release factor will come in and bind in the A site, and that binding between the tRNA and the P site and the last amino acid of that peptidyl chain will become weakened and will break. And so the protein is released. And that's going to cause the translation complex to just disassociate. And so let me show you that picture. And so again, this is just called termination. So again, the ribosome gets to a stop codon, either UAG, UAA, or UGA. All 3 of these codons code for stop. And so into the A site will come the release factor. Once this binds, that's going to cause the amino acid tRNA here in the P site to release the protein. And so all these little blue circles here represent individual amino acids that have been put together during this process. this will be released. And so that's shown here. We have a free polypeptide, and that's going to cause the rest of this complex to come apart. Okay, and that's it. Just going to mention a few more things, and there's actually a typo on this slide. So in both prokaryotes and eukaryotes, you could end up with what's called a polyribosome. In this particular example, and on the next slide, I'm going to specifically talk about prokaryotes. But I do want to just mention that in eukaryotes, you can have a polyribosome as well. So what is this? So you can see this is an mRNA molecule right here in orange. And you've got one ribosome that's already finished translating it, and it's already released its polypeptide. You've got a second ribosome that's already bound to it before the first ribosome's even done. It's getting close to the end, and now you have a third ribosome starting the process. And so multiple ribosomes can attach to mRNA and read it kind of simultaneously, which speeds up protein synthesis, right? Now, what I do want to mention here, a difference between prokaryotes and eukaryotes, is that in eukaryotes, translation of mRNA can't happen until after the mRNA exits the nucleus. So transcription has to be totally complete. The mRNA transcript has to be processed and then moved to the cytoplasm before ribosomes could start to do their work. Where in prokaryotes, that's not the case, because transcription happens, essentially in the cytoplasm, right, in the nucleoid region, but there's no membrane around that DNA. And so the process of transcription and translation in prokaryotic organisms can actually happen simultaneously. So as the RNA transcript, the mRNA transcript comes off of the DNA, ribosomes can just start to bind. And so again, you'll see polyribosomes. So the figure on the next slide actually shows this a little bit better. Okay, so again, in a bacterial cell, there is no nucleus, and the DNA is actually circular. It's not shown circular here. It's still double-stranded. You can get transcription and translation at the same time. And so that's really shown here. So in blue, we have DNA. These yellow spheres are the RNA polymerase. I'll just show one of them for a second. So RNA polymerase is reading the gene. As it reads the gene, it makes this pink mRNA transcript. And then these little pink ribosomes attach and start to read it. As soon as the 5' end comes off, these can bind and start to read. The purple dots are the amino acids being put together. And so again, a polyribosome is when you have an mRNA transcript that's being read by multiple ribosomes at the same time. And so look at all the proteins that are being produced all at the same time from the same mRNA molecule. Makes it a very efficient process. So the very last thing that I want to leave you with is after these proteins dissociate from the ribosome, they have to be folded, right? And so in order for a protein to function, it has to be in its proper three-dimensional shape, right? Form equals function. And so oftentimes there are these specialized ribosome-associated proteins called molecular chaperones, that's the title on the slide, that actually help facilitate this process, okay? So they will create a space and assist proteins in this folding. Without molecular chaperones, there could be various regions of the amino acid chain that might interact inappropriately and prevent the proper folding process from occurring. And so again, molecular chaperones ensure proper three-dimensional shape. Molecular chaperones may also be involved in the cleavage of proteins, so the cutting of a section or two sections, or putting proteins, protein parts together. So proteins can be not only folded, but they can be modified after they're made. Okay? And so that's what I wanted to leave you with. That's the end of this chapter.